Enzymatic synthesis of high-density RNA microarray.
Hi and Assalamualaikum. My name is Nur Shuhaza Izzarin binti Muhammad Nizar. In this presentation, my group will be discuss the article about microarrays. So, not all people know about microarrays and some people might be wondering what is microarrays, right? So, a microarray is a laboratory tool used to detect the expression of thousands of genes at the same time. This also consists of probes that demonstrating several different genes organized on a glass slide in a systematic pattern. Now we know about microarrays, but what is the function or the objective of people use microarray? So the function or the objective of microarray is to determine whether the DNA from a particular individual contains a mutation in genes. And for the information, the origin of DNA is that DNA originated from RNA in an RNA or protein environment. There are so many things that exist in this world and for sure have something that we can use in our daily life. Then, how can we apply the microarray in our life? Microarrays have many applications and mainly used in the field of medicine and related areas. If it was used in the medical areas, it can be categorized into 4 types, which are the discovery of the target that microarray use to compare the diseased tissue or cell with the healthy tissue to find the characteristic of particular disease. The next one is the discovery of drugs and leads that microarray use to screen potential compound and identify the toxicity of the lead compound. Next is in diagnostic and prognostic. Microarray used to know the state of disease, type of tumor and other factors important to the patient. Let's say in pharmacogenomic and theranostic, the microarray used to decide a patient treatment and therapy on the basis genetic makeup. So not in medical field only, microarray also been used in the field of agriculture to studies about pest control. In forensic analysis, it used to get the detail of DNA that will helpful for investigation purpose and many more fields that use microarray. In this article about microarray, there are basic protocol and protocol 1, protocol 2. Basic protocol is about conversion of a DNA microarray to an RNA microarray. Protocol 1 is the detection of RNA via hybridization, and protocol 2 is about RNase HAC. Now, before we go deeper in the step of basic protocol, a little bit of information for everyone that there are some steps in basic protocol that also been used in protocol 1 and protocol 2. For the example, in the preparation of reagents and workplace, step 1 until 5. In hybridization, step 7, 9 until 17 were used in protocol 1. And in stringency washes, step 21 until 26 also been used in protocol 1. For protocol 2, the step 3, 4, 5, 7, 8 and 9 is the same step as in basic protocol. Now we will move to the step of basic protocol. We will start with the preparation of reagents and workplace. First, the liquids of the washing buffer NSW and SWB with 30 ml each in 50 ml reagent tubes has been prepared. Then we continue with the liquid that has been prepared and stored at room temperature in the dark until it used. Lastly, all the surfaces and pipette has been cleaned with RNase away.
Next, let's move to the next step, which is the hybridization of primer. An RNase-free 1.5 ml microcentrifuge tube are prepared and lacerated BSE and primer oligonucleotide are led to thaw at room temperature. The hybridization oven was switched on and the temperature are set to 37 degrees. The hybridization solution are prepared by mixing the reagents according to table number 1 in a sterile 1.5 ml microcentrifuge tube. The glass slide with the DNA template microarray was placed on the bench. At this step, ensure the side of DNA is facing up. Next, a hybridization chamber was dipped with the Christ size and tweezer was used to remove the thin plastic foil, and the adhesive on the rubber gasket was exposed. The hybridization chamber was placed over the DNA array and stick to the glass surface, then were fixed by tightly pressing on the rubber gasket. Then, the hybridization solution was mixed by flipping the tube carefully. The hybridization buffer contains twin 20, resulting in bubble formation upon excessive mixing. The presence of bubbles can complicate the application of the solution in the following step. Next, the hybridization solution was carefully added to chamber with a pipette through one of the holes of the hybridization chamber. Second last of the step, the holes are sealed with the adhesive seal tapes. The slides are briefly rotated to ensure proper mixing is facilitated by movement of the air bubble. Last step is the slide are wrapped with aluminum foil and was placed in the hybridization oven and incubated for 30 minutes at 37 degrees with rotation. Some of these steps are also used in hybridization of RNA hybridization by using fluorescently and different reagent solutions to mix. RNA hybridization are used to assess the success of conversion process.
Next, we look at cross-linking method. The UV light source was placed at 4 degrees about 30 minutes to equilibrate the whole device. Then, the UV light intensity was measured using intensity meter in 60 seconds. In 1.5 ml microcentrifuge tube, 2 times MES hybridization buffer with 1:1 ratio was mixed with RNA's free water. The slide was placed on the bench with the DNA array is facing up. Next, thin plastic foil was removed to expose the adhesive on the rubber gasket. The hybridization chamber was placed over the DNA array and stick it to the glass surface. 1 time MES hybridization buffer was applied to completely fill the chamber. After that, the whole of the reaction chamber was sealed with adhesive seal tape. Then, the glass slide was placed above the UV light source with hybridization chamber with facing upward. Next, pre-incubation solution was prepared as shown in the table 3. Then the power supply was unplugged to switch off the UV light. The adhesive seal tab was removed carefully from hybridization chamber with the tweezer. Next, the buffer was pipetted out and discarded. After that, 1 volume of RNA's free water was pipetted in and out of the hybridization chamber about 30 seconds, and then the water was discarded. The last step in cross-linking, where the above step was repeated for another 30 seconds.
Next method is pre-incubation method. Apply the pre-incubation solution to the hybridization chamber. Seal the whole of chamber with adhesive seal tabs. Then, wrap the slide in aluminum foil. Incubate the hybridization chamber at 37 degrees Celsius for 30 minutes with rotation in the hybridization oven. In the meantime, prepare the primer extension reaction solution in a sterile microcentrifuge tube at room temperature. Remove the adhesive seal tab from the hybridization chamber. Pipette out the solution and discard it. Primer extension method. The first step is add T7 RNA polymerase to the primer extension reaction solutions. Mix the solution carefully. Then apply the solution to the hybridization chamber. Seal the hole with the adhesive seal tabs and test for movement of the solution by rotating the slide manually. Wrap the slide in aluminum foil. Then incubate the chamber at 37 degrees Celsius with rotation for 60 hours or overnight in the hybridization oven. At the end of the incubation time, prepare the reaction mix for the DNA template degradation in a sterile microcentrifuge tube at room temperature. Remove the adhesive seal tabs. And lastly, pipette out the solution and discard it.
The next step is the syringes washes. Firstly, the adhesive seal tabs was carefully removed. Then the hybridization solution was pipetted out and it was discarded. And the hybridization chamber was removed with the tweezers. Then the slide was placed into the 50 ml tube in 30 ml of NSWB and shake vigorously for 1 minute. Next, residual buffer on the walls of the reagent tube was rinsed off to prevent carryover into the next solution. The slide was placed in the 50 ml reagent tube with 30 ml of SWB and shake vigorously for 30 seconds. And the step 4 was repeated, and the slide was placed into the 50 ml reagent tube with 30 ml of one-time FWB and shake for 5 seconds. Make sure that no soap deposit is left in the array. Lastly, the slide was placed in a microarray centrifuge and spin dry for 30 seconds.
The first step of turbo DNase reaction is turbodinase was added to reaction mixture and mix it well by pipetting up and down. The next step is reaction mixture of DNA template degradation was applied to the hybridization chamber. Next step is holes was sealed with adhesive seal tabs and the slide was wrapped with aluminum foil. The next step is incubate at 37 degrees Celsius with rotation for 5 hours in the hybridization oven and pipette out the solution and discard it. Other step is 1 chamber volume of NSWB buffer was taken, pipette it in and out of the chamber and then discard the solution. The last step is the slide of reagent tube was placed with 30 ml of one-time FWB, then shake for 5 seconds, and lastly, the slide was placed in microarray centrifuge and spin for 30 seconds.
Lastly is the conclusion in this article. In this conclusion, there are some expected results from the article. Firstly, the hybridization with a fluorescently labeled complementary probe can detect the presence of the specific ssRNA sequence. Next, different RNA strands with its characteristic of oligonucleotides such as length, sequence modifications, and sequence context of label can be identified in comparing the results. And next, the residual RNA was detected via hybridization with the CY3 label DNA probes template X and template Y. And lastly, the incubation in the absence of RNase H also induced loss of RNA from the surface during 2 consecutive hybridization steps to the same RNA sequence performed.